We are starting with Morula to Blastula transition in case of early development, and we will be talking about this development in mammals, and we will also talk about human development as well. So what are the characteristic features of early Morula? How can we identify this stage? So early Morula is 8 to 16 cell stage, or in some species it can be either 8 cell stage or 16 cell stage. It can go up to 64 in some species. For humans, it is the third day of development when this early Morula stage will develop. And in case of humans, typically 8 cell stage is termed as Morula, and it is formed after third to fourth cleavage division of zygote. What are the characteristic features of this early Morula stage? I am talking about this 8-celled stage of Morula. So as you can see, the outer membrane zona pellucida is present, and there is loose mass of cells, or we can say loose spherical mass of cells. It is spherical shape, and each and every cell of these 8 cells is totipotent. By totipotency, we mean that they can give rise to entire human body as well as sportive tissue. For humans, sportive tissue usually include tissue like placenta and some other extra embryonic tissue as well. For this early Morula, it has to be noted that the cells are entirely dependent on maternal mRNA and proteins. The entire genome of these cells is suppressed. As we saw in the video of cleavage, there is only S phase and M phase active. The G1 phase and G2 phase are entirely suppressed or very much reduced. So these cells are entirely dependent on maternal factors, the maternal mRNA and maternal proteins. These cells right now cannot make their own mRNA and their own proteins. But around 8 cell stage, or after the 8 cell stage, the genome activation will take place, and these cells will start producing their own mRNA and their own proteins for the very first time. This 8 cell stage of Morula will undergo compaction. Now compaction is the process by which this loose mass of cells will become an arranged or compact structure, which will ultimately lead to formation of a late Morula and respective Blastula. Now this compaction process will start with 8 cell stage in case of humans, and it may vary with the variation of species. This compaction is mediated by specific cell adhesion molecules, and in this case these cells will start expressing a specific cell adhesion molecule which is known as epithelial cadherin. And this epithelial cadherin or E-cadherin is calcium dependent. It is attached to the cytoskeleton, specifically to actin cytoskeleton via beta catenin and alpha catenin proteins respectively. As you can see in this diagram, this is the structure of cell membrane of these cells, and they will start expressing these cadherin proteins. Now there are having cadherin domains, and there is specific calcium binding site because these are calcium dependent proteins. If we look at cytosolic side, these cadherin proteins are associated with actin cytoskeleton through catenin proteins, and they are P120 catenin, which is protein 120 catenin, there is beta catenin and alpha catenin respectively. Some of the cells of early Morula will acquire apicobasal polarity, and these cells will be acting as peripheral cells, and some of the cells will not acquire this polarity, and those cells will act as the cells of inner cell mass. This 8 cell stage will undergo the zygotic genome activation, which means that these cells will start producing their own mRNA and their own proteins for very first time. When this zygotic genome activation will take place, there will be differential gene expression and first lineage decision will be made. The first lineage decision is that which cell will act as the outer cell, or which cell will make this trophectoderm. In humans it is known as trophoblast, and which cells will act as the inner cell mass. Now it has to be noted that both of these, inner cell mass as well as outer cells, will lose the totipotency. Whereas in case of 8 cell stage of Morula, each and every cell is totipotent. Each and every cell will be able to give rise to entire human body as well as its supportive tissue. Whereas in case of outer cells will become multipotent because they will give rise to supportive tissue like placenta, whereas inner cell mass will become pluripotent, which means that inner cell mass can give rise to entire body, but they cannot form the supportive tissue like placenta. In this figure, you can see that two cells are joined together by these Cadherins, and these Cadherins are calcium dependent. There are calcium binding sites. We talked about these calcium binding sites in this diagram as well. On cytosolic domains, these are attached to this actin cytoskeleton by this beta catenin and alpha catenin respectively, and the cadherins of two cells will interact to form tight junction like structures, and these cells become compact. That is the whole point of this process of compaction in this morula. Loose cell mass will become this tightly packed arranged structure. Let's talk about some features of these peripheral cells as well as inner cell mass. For peripheral cells, there will be higher expression of cadherins, which are homophilic interactions and calcium dependent adhesion interactions. They will form tight junction in case of peripheral cells as well as desmosomes, and these tight junctions will make a epithelial like barrier. They will also express the integrin, which are another type of cell adhesion molecules, and these integrins will be responsible for interactions with extracellular matrix. Means that these integrins will help in the process of implantation of embryo. Now, these peripheral cells are the ones which will acquire apicobasal polarity, which means that these cells will have a distinct apical side and a distinct basal side. And on apical side, there will be distinct villus structures, or we can say microvilli structure as well. In case of inner cell mass, there will be expression of cadherins, but interactions will be trans interaction. In case of peripheral cells, there were tight junctions, but in case of inner cell mass, there will be no such tight junctions. Instead, there will be gap junctions in case of inner cell mass. The cells of inner cell mass will not acquire any apicobasal polarity. This table represents the basic differences between these outer cells and inner cells. These outer cells will express these strong homophilic cadherin interactions, whereas in case of inner cells there will be cadherin but weaker interactions due to only trans contacts. In case of peripheral cells, there will be tight junctions and desmosomes. In case of inner cells, there will be gap junctions. In case of peripheral cells, there will be specific apicobasal polarity. No such polarity will occur in case of inner cell mass. Talking about secondary cell adhesion molecules, there will be integrins, which will be associated with extracellular matrix, and they help in the process of implantation. In case of inner cell mass, there will be connexins. And what will be the fate of these outer and inner cells? The outer cell will form Trophectoderm, which is called trophoblast in case of humans, and in case of inner cells, it will form inner cell mass. Talking about summary of these outer and inner cells, the outer cells have cadherins and tight junction proteins like claudins and occludins. There are desmosomes which stabilize the epithelial sheet. In case of inner cells, there is mainly cadherin and the gap junction connexins like CX43. In case of late stage of morula, the cells have formed a compact mass. It can be 16 or 32 cell stage, and in humans it is around day 4 of development. Compaction has completed, means there are tight adherent blastomeres. In case of cells of periphery, there are tight junctions, and in case of cells of interior, there will be gap junctions. And zona pellucida is still intact, which means that this solid mass cannot undergo the process of implantation yet. Zygotic gene activation is also completed, which means that outer cells are flattened, acquired apicobasal polarity, acquired tight junctions, desmosomes, and have lost totipotency. For inner cells, they have acquired gap junctions, they have lost totipotency, but they have not acquired any polarity like outer cells. In case of outer cells, there will be expression of specific genes which will bring their multipotent fate and help them synthesize the supportive tissue like placenta. The two of the most important genes are CDX2 as well as GATA3, and these expression of genes will restrict the lineage of these outer cells to placenta as well as extraembryonic tissue. It is multipotent fate of these the trophoblast cells. In case of inner cells, we have expression of OCT4, NANOG and SOX2, which will bring about the pluripotent fate, and these inner cell mass cells can give rise to entire body but cannot give rise to this placenta as well as extra embryonic tissue. Though some of the extraembryonic tissue like some part of yolk sac as well as amnion is still derived from this inner cell mass. Now we will talk about the process of cavitation. When blastula formation takes place, there will be formation of a water filled cavity within the compact morula, and at this stage the morula will be called blastocyst. In humans, it will occur around day 4 or day 5 of development. This is the structure of blastula. We have still zona pellucida intact. We have outer cell mass which is termed as trophectoderm. In humans, it is called trophoblast. This is inner cell mass, and there is a water filled cavity which is termed as blastocele, and at this stage this morula is known as blastula. Now, how this cavitation process takes place, we will see in this slide. So there are sodium potassium ATPases, or we can say sodium potassium pumps, present on these peripheral cells, on these trophectoderm or trophoblast cells. So let's have a look at this trophoblast cell. This is the apical surface, this is zona pellucida, this layer, and we will be seeing one of these cells. This is apical surface, this is basal surface. On the basolateral surface there are sodium potassium ATPases. Now these sodium potassium ATPases will mediate the efflux of sodium ions. Sodium ions will move out of the cell, and there will be influx of potassium ions, means potassium ions will move into the cytosol. What is the role of this sodium potassium ATPase? The role of this sodium potassium ATPase is to push out the maximum amount of sodium out of the cell, and when sodium is pushed out of the cell, there will be formation of gradient of sodium. The sodium will be present in higher concentration outside the cell as compared to inside of the cell, and wherever solute goes, water follows it. Wherever sodium goes, water follows it. So water from the cell will start coming out through these specific aquaporins, which are aquaporin 3 and 8 present on the basal surface of these trophoblast cells. But what is the primary source of this water? Is it the cytosol of this trophoblast cell, or is it some other kind of source? So the primary source of this water is the uterine environment. The uterine environment will start pushing some of its water content into these trophoblast cells through specific aquaporins which are present on the apical surface, and these are aquaporin 9 and aquaporin 8. So uterine environment will start pushing some fluid to these specific cells, to aquaporin 9 and 8 present on apical surface, and the sodium potassium ATPase is working, which is developing the gradient of sodium ions. The sodium ion concentration will rise outside the cell. Water follows the sodium ion, and the water will start leaking out of these trophoblast cells, and this water will follow sodium ions. There will be hydrostatic pressure, and this hydrostatic pressure will result in the formation of small cavities. There will be multiple small cavities which will be forming, and these small cavities will unite together to form this large cavity which is called blastocele. As you can see, the morula, and there is formation of blastocele cavity which is primarily mediated by this sodium potassium ATPase, and primary source of water is this uterine fluid. What will be the fate of inner cell mass or trophectoderm, which is outer cell mass? In humans it is called trophoblast. So how can we say that this trophoblast will form specific tissue and this inner cell mass will form entire body? We can say that what decides the multipotency or pluripotency of these respective cells? let's have a look at this blastomere of morula of 8 cell stage. Whenever there is suppression of zygotic genome, means this cell cannot synthesize its own proteins, there will be the presence of OCT4 mRNA. This OCT4 mRNA is maternal in origin. It came from egg itself, and whenever the 8 cell stage morula will undergo its zygotic genome activation, it will start forming its own OCT4. So OCT4 concentration will be much higher, and along with the OCT4 it will start forming CDX2 as well. Now OCT4 as well as CDX2 are mutual inhibitors of each other. OCT4 inhibits CDX2 and CDX2 inhibits OCT4. Now these two genes will be forming two different fates of different cells of this blastula stage. Now let's have a look at inner cell mass blastomere. In case of inner cell mass blastomere, the OCT4 expression will dominate. Now this OCT4 expression will bring about the expression of two other genes, which are SOX2 and NANOG, and this OCT4 upregulates the expression of SOX2 and NANOG, and they upregulate expression of each other. So this OCT4 will decide the pluripotent fate of the inner cell mass blastomeres. But something different happens in case of the outer cell mass or trophoblast cells. Let's have a look at trophoblast cell. So this is the trophoblast cell, and we are looking inside the nucleus where there are genes for the synthesis of this CDX2. In case of outer cell mass, there is a specific protein which is TEAD4 and activator of TEAD4 which is YAP protein. These two proteins will bring out the expression of CDX2, and CDX2 will maintain the Trophectoderm identity, and finally we activating some other genes which will ultimately bring about the fate of supportive tissue. These other specific genes are EOMs and GATA3. The EOMs and GATA3 will bring about the epithelial polarity as well as specific proteins which are required for implantation process. TEAD4 will bring about the expression of CDX2, which will bring the expression of EOMs, PSX1, HAND1, which are all trophoblast specific genes, and they help in the formation of supportive tissue as well as implantation, and they give identity to these trophoblast cells. In case of OCT4 cells, there are some specific hippoproteins which are very much active. Now it has to be noted that these hippoproteins are inactive in case of trophoblast cells. They are not active. But in case of inner cell mass, these hippoproteins are very much active, and they give rise to this LATS protein, which is a type of kinase, and this LATS protein will be phosphorylating the YAP protein, and if this YAP protein is phosphorylated, it will not activate the TEAD4 protein. And if TEAD4 protein is not activated, expression of CDX2 will not be there, and there will be no CDX2 protein in case of inner cell mass. That's why in case of inner cell mass there will be OCT4 activation only, and there will be expression of OCT4 mediated genes which are OCT4, SOX2 and NANOG. Whereas in case of this outer cell mass or trophoblast, the OCT4 expression cannot take place, because if there is CDX2 expression, this CDX2 will inhibit the expression of OCT4. That's how these outer trophoblast cells and this inner cell mass will gain their respective identities, and this inner cell mass give rise to whole body, whereas this trophoblast will give rise to specific supportive tissue only.